First of all, apologies for having to cancel class and lab today. You can blame this one right here. She has a fever, and so she can't go to daycare. Probably something microbial going on right now with her, right? So microbes can impact us directly or indirectly in this case. So before I start, feel free to use the chat if you want. I try to keep my eye on that. If not, just shout out some questions or comments and we'll do it that way. I'll go over the slide deck that I've created and then kind of talk about these bulleted aspects and kind of what I want you to know involving that material. So before we start, are there any specific questions you have or specific aspects of the chapter that you want to go over first? Maybe not. Okay, well, in that case, let's just get started at the beginning, okay? Let me get my share my screen. It's been a while since I've had to do this Zoom business. There we go. Okay, so hopefully everyone can see my slide deck and maybe give me a thumbs up or something, icon or in the chat or shout out if you want that you can see it, just so I don't, there we go. Okay, thanks, Paula. Let's get started. So what I did was I just compiled all of the slide decks and I cut out everything that I feel like is not going to be necessary for the midterm. And so that chunked it down to 194 slides, I think, something like that. So that may seem like a lot, but it was a lot more before that. Okay, and so I've left the cover slide for each chapter so you kind of know where it starts and stops. So with the first chapter, it says define microbiology. That's just simply the study of microbes, right? Ology is study, micro is small. So study of things that we can't see without some kind of magnification. And then be familiar with the different classes of microorganisms, right? So we will talk about all of these. We've really only talked about bacteria for the most part, but we will get into viruses, fungi, protozoa, helminths, and algae in time, okay? And then there's also Apicomplexa, which is the malaria carrying parasite. That one used to be under protozoa, but has been classified in its own category now, okay? Know the difference between a hypothesis and a theory, right? If something is just a theory, that doesn't make sense, right? A theory is something that is very well supported independently by multiple different scientific investigations and inquiries from different scientists. Whereas a hypothesis is, an educated guess, right? And so be somewhat familiar with these concepts, right? Principle, law, theory, hypothesis, they're all somewhat different, meaning different degrees of confidence in the validity behind them, right? Let me make this smaller here so you can see the whole screen. Oh, hold on, I think let me pin myself. There we go, getting back into it, brushing the cobwebs off. Okay, be familiar with this flow, right? Okay, you might see something like this on the midterm for like a label and diagram, for instance, where know this process. Observation, right? My daughter felt warm this morning when I got her out of the crib. 2, question. I wonder if she's sick. 3, hypothesis. I think she's warm because she is sick, and that is giving her a fever. Experiment. I'm going to get out a thermometer and I'm going to collect data. Analysis. She has a temperature of 100.5. Interpretation. She is sick. Conclusion. She can't go to daycare. And now I have to cancel my class. All right. So be familiar with that. There we go. So, right, a scientific theory has little or no evidence to support it. Best guess. That is wrong. That a theory has a lot of evidence and is not a guess. Okay. Why is this guy on here? I thought I got rid of those guys. Robert Hooke, of course. I got rid of a lot of the old dead white dudes. Robert Hooke was the one who first came up with the name cell, looking at cork under the microscope, and Van Leeuwenhoek was the first to really look at living microbes under a microscope. So then a couple other bullets, how old the Earth is and how long bacteria have existed, right? So we know the Earth is 4.5 billion years old, and we know that microbes have been around for 3.5 billion years of that Earth's existence. So the Earth was really only around for 1 billion years before microbes came around. Okay. Know the difference between prokaryotes and eukaryotes, right? They're both cells. They can both be single cells. They both have similar structures in terms of membranes and ribosomes and actin, right? How they differ is that no nucleus, no organelles. Eukaryotes, you do have those things, right? So they have similarities and differences, and you'll want to know those differences, okay? Be familiar with some of the top worldwide causes of microbial death, why they differ from neglected tropical diseases. Okay, so there should be some tables in there. You can consult the tables if you want to, but things like influenza is going to be a worldwide cause of death. It's going to be different from things like malaria and certain helminth infestations, simply because influenza impacts all of the world, whereas something like malaria only affects primarily people in the tropical regions. And so those are neglected tropical diseases. Okay, that's really all you need to know, right? An example of a worldwide pathogen and an example of a neglected tropical disease. What you went down. Steps of the scientific method, covered that, and the levels of taxonomic classification, right? So domain, and then kingdom, phylum, class, order, family, genus, species. That's it. Oh, okay, I'm going to put her down. You're going to go play around. So here's some taxonomy. Again, you don't need to know anything about Carl von Linné. Really just be familiar with that taxonomic order, right? Genus, capitalized, species, lowercase. I do hope that you know that just as we name certain bacteria from here on out, that's going to be the way we do it. If we can't italicize, then we just underline those, okay? Any questions so far? No? Okay. Oops, I think I got away from my screen now. I gotta find the right, where did that go? Oh, here it is. Okay. What else do we have in chapter 1? Here's the taxonomy, right? Mainly, the only difference that we talked about here is how Don't worry about these characters that did this, but how the taxonomy expanded over time from just animals and plants to then protists and Monera being added, and then finally fungi. Okay. These are some terms that I think you're probably pretty well familiar with by now. Here's again the life. You don't need to worry about all these organisms and where they fall on the timeline of Earth. Just know that the Earth is 4.5 billion years, or specifically know that microbes are here starting 3.5 billion years ago. Okay. Prokaryotes and eukaryotes, right? Those are the differences and similarities. There are also, of course, viruses that are acellular. They're not living. Prions, acellular, not living. Okay. And then some terms from this, chapter 1, kind of bleached out, but right here, the terms, right? Pathogens, microbes that do harm. Emerging disease, a newly identified pathogen, right? So COVID is caused by SARS-CoV-2. That's an emerging disease. And we covered this actually in the last slide, chapter as well. So kind of bringing this back again. Zoonoses is not an infection that specifically happens in the zoo, right? Sure, it could, but that is not what a zoonotic infection is. It is an infectious disease that comes from an animal, right? Whereas re-emerging diseases have already been around. They come and go. The plague is one. Measles is now one coming back. Smallpox, we're seeing that re-emerge. You spilled all your cereal. So some terms there. Theory of biogenesis, right? That was at the beginning here. That basically put the final nail on the coffin of spontaneous generation, that things can come from unliving, like life comes from unliving matter based on these vital forces. Whereas theory of biogenesis is that no, if you're alive, you came from something else that was also alive. Okay. Law, theory, hypothesis. All right, I think that covers chapter 1. Any questions on chapter 1 that I haven't addressed? What? Don't see anything in the chat. Yeah, we good? Okay. You good? You ready to move on? You want to move on to the next chapter? Start them young. No. No, you don't? Well, we're going to. All right. So chapter 2. The 6 eyes. Know the 6 eyes. You know, that might be a good short answer or essay question, right? Use the 6 eyes to diagnose some kind of infection in somebody. Or, what would you do if you were wanting to know if there was a certain microbe somewhere, right? What's the story? You don't need to memorize this table. You need to memorize the order, of course, but what are you doing in that process? And we've done the first 3 steps in lab, right? Inoculation, right? We've taken a sample, a swab from something, and we put it in media and we grow it. Incubation, right? We grow it at a certain temperature, either at 30 degrees or 37 degrees. Isolation. We take that and we spread it on a plate. We do streaking. We try to get individual colonies to isolate the microbes. That's where we've stopped. Today, we would have done inspection. We would have looked at our plates and said, did I get separate colonies, right? What does it look like? What do those colonies look like? You want to go in your high chair? Do those colonies all look the same? Like, could it be just the same bacteria? Do the colonies look different? Do I have some that have different borders and different shapes and different colors, right? Then information gathering. And that's what we're going to do later this week. We're going to do stains, right? What does the shape look like? Is it streptococcus, right? A strip of circular bacteria. Is it bacillus? Is it staph, right? And then identification. Well, based on the shape and the fact that it's gram negative and it has these colony morphology characteristics, I'm going to say that the bacterium must be E. coli. Now what's wrong? You went out? Sorry, folks. This is life with a 1-year-old. There you go. Go play. Go do something. All right. So those are the 6 eyes. Be familiar with the 6 eyes. Okay. Wink, wink. Know the purpose of immersion oil, right? Here are the steps. We don't need to go through these. Purpose of immersion oil is to improve resolving power or the ability to show detail, right? So the 2 major things are magnification and resolving power when we talk about microscopy, and those are the terms and definitions section of chapter 2, right? Magnification makes it bigger. It doesn't necessarily make it clearer, right? It just takes something small and makes it big. So you need resolving power to take that small thing that's been made big to make it clear. Yeah, get that out. Coughing, that is a sign, right? Not a symptom. How do we find the total magnification? Well, this was a quiz question, right? You take the objective power times the ocular power. So I think for the quiz, it was, how do you reach a magnification of 400x if you have a 40x objective? Well, you use an ocular that has a 10x, right? Because 40 times 10 is 400. Resolution just gives us more detail. And the purpose of immersion oil, so to specifically address that bullet, that second bullet on chapter 2, immersion oil has the same refractive index as glass. So the light doesn't bend wonky when it goes from glass to air to glass. It's just going from glass to immersion oil with the same refractive index to glass. And now it's not bent and you don't lose information that way. So the more information you get, the more clear that image is going to be. All right. Different types of microscopy, right? Be able to differentiate these. Bright field. The field is not what the specimen is. The field is what the specimens are on, right? Bright field is a soccer field and the bacteria are the soccer players. The field is light. It is lighter than the specimen. Whereas dark field is the opposite, right? Dark field is dark and the specimens are light and they stand out from that dark background. Phase contrast, that's the technique that they used at Stupid to look at those microbes in the water, right? Because they're clear. You can do bright field, but you won't see that much because the microbes are clear. Oh, you dropped them again. The microbes are clear just like the slide. Okay, you're welcome. So we use phase contrast to subtly take those waves and bring them out of phase from each other. And that allows it to stagger and see images that are clear. And this is good for live cell imaging, right? That's why they did that at Stupid. You don't have to kill them to stain them. Right? We are going to kill bacteria this week and stain them. You can't see how they move or if they move if you kill them, right? Fluorescence microscopy, right? We use fluorescent dyes and we shoot it with UV and that causes it to fluoresce. That's nice because we can individually label different structures and proteins to see where they are in that cell. I wouldn't worry about the fluorochrome and the primary and secondary antibody and the antigen. Don't worry about that stuff. That's how it's done, of course. But you don't need to know those specific steps. Okay. Just kind of like on the quiz, right? There was that matching. It'll be that level of detail. Just know the difference between these few different techniques. Okay. Electron microscope, that's going to give us the most resolution. And so the highest magnification power. So we can look at viruses that way. Okay. What else? The different staining techniques, right? So wet mounts, you guys made wet mounts in the lab. Fixed mounts is what we will do in lab this week. We will take some bacteria in the broth, put it on the slide, and pass it over the Bunsen burner, and that will dry up the media, and it will also kill the bacteria, and then they will stick to the slide, so then we can put a stain on them, okay? There's also many different stains to choose from, right? And be familiar with these, right? Okay, know that a basic dye uses cationic positively charged chromophore, okay? Cationic, positive, right? So basic dyes are positive staining. We stain the surface of the microbes, they're negative. Opposites attract, okay? So this is methylene blue. We're gonna do that this week. It's a positive stain using a basic dye. Whereas acidic dyes are the opposite. Acidic are negative, anionic. Okay, so the microbes with that negative charge are going to repel the dye because opposites attract, same repel. And so here's an example of India ink to look at capsule staining, for instance. All right. You don't stain the microbe, you stain the background. All right. You stain the field. That doesn't make this dark field, but similar property, whereas what you're looking at is the contrast between a dark background. Okay. Simple stains, differential, structural, know these differences. A simple stain just uses one dye. That's what we're going to use this week, methylene blue. Whereas differential stains use a primary stain and a counter stain. That's going to be like the Gram stain, which we'll also do soon. All right, you stain it with the crystal violet. If it's Gram positive, it's going to take up that crystal violet and it's going to fix it with the alcohol, right? Or the iodine, and you're going to wash it away if it doesn't. And you're going to counter stain with Safran O. And so it's going to be differential. It's going to be one color or the other color. And that's going to tell you something. Gram positive or Gram negative. And then structural stains are going to give us different parts of the bacteria. What's wrong? Are you melting down? What's going on? You want to go in your chair? I think you have a stinky diaper. So structural stains are going to be like capsule stain with the India ink, right? You can stain different things like flagella or pilus, Fimbriae, right, to give us information on what structures those bacteria have. Here's simple and negative, right? So on the left, we have methylene blue. That's a positive stain. On the right, we have a negative stain, right? Depending on what's being stained and what isn't. You want to sit in here? There you go. No, you don't want to sit in there. Differential stain, right? Gram positive on the left side, Gram negative on the right side. Acid fast is another example. We will get into, we'll do acid fast later, but that's used for for staining cells that don't stain Gram positive or negative, right? Particularly like tuberculosis, mycoplasmas. Okay. Any questions? I haven't noticed any chat or anything like that. So I'm going to keep moving along. I'm assuming people are still here. Yes, I see that they're here. I'm going to still move on, but feel free to interrupt me at any point if you have clarification that you need on this. Okay. All right. More differential stain, structural stain. There's the capsule stain. Oh, here's a chat. I see it just popped up. Should we know the examples for each staining method? I think it wouldn't hurt to know that for instance, a negative stain is using a capsule stain. A positive stain is methylene blue. A differential stain would be gram positive. That wouldn't hurt, but I think the most important thing is to be able to know the difference between those, right? Examples are icing on the cake. What do you want? You want this? Good question. Thank you. No, you don't want that? All right. Different types of media, right? So we have synthetic. We know everything that's in synthetic media, right? Because we've made it. Whereas complex is non-synthetic. We know what some of the stuff is in there, but we don't know everything that's in there, right? And then we have general purpose. That's going to be your nutrient auger, your nutrient broth, right? General purpose grows a broad range of stuff. Whereas enriched media, that is going to be for the picky eaters out there. Oh, you stuck, right? The picky eaters that like blood or serum, or certain growth factors or vitamins. So they will have enriched media. There you go. Okay. Selective media, differential media, right? And some can be both. If it's selective, that means it's going to grow something or not, right? It selects for growth of bacteria. Differential says, okay, well, I'm going to grow both of those things, but based on the growth, you'll be able to tell if it's this or that, right? So different colors, right? Not just there or not there. It's there and it's red or it's green, right? So some media can be both. It can be differential and selective. What else? Okay. One other thing, it says ocular versus virtual image and real image. So the real image, of course, is what is being transferred into that lens of the microscope. But if you remember, there was one slide where we talked about how it actually flips it upside down. So what you see is the virtual image. What you see is that image flipped upside down. So if it looks like this is the real image, you're seeing it like this. All right. That's all you need to know about virtual and real image, that they're flipped. Okay, any questions on chapter 2 that I did not cover? Remember, if questions do come up, shoot me a message between now and the end of Tuesday, right? If you're shooting me messages Wednesday morning, it's probably going to be too late. But between now, I'm stuck at home with a cranky 1-year-old. So shoot me some messages if you have questions. What else am I going to do? All right. Chapter 3. Be familiar with the different characteristics of life. Okay. So where is that? Did I get rid of that? I must have. Okay, interesting. Characteristics of life are going to be, oh, well, here's some characteristics, right? Shape. They're going to be, they need to be metabolic, right? They need to undergo metabolism of some sort. They have to have heredity, right? DNA or RNA that they pass on to their babies. Okay. They need to have some kind of cell that surrounds all of their good stuff, right? Their DNA, their ribosomes, the cell, the membrane that protects it and allows it to go undergo those metabolic processes. Those are the characteristics. So it's not going to be a long list. Know some of the general things that make cells alive. What? What else? Internal, we've already said know that, be able to compare and contrast ukes and prokes. This was on the quiz. It could be on the midterm. No promises, but there's a good chance it might be. So be familiar with these, okay? Know these different structures, okay? If during the test you're not sure what it's pointing to, well, ask me. If you're not there to ask me, well, give it your best go, right? Don't give it a theory, give it a hypothesis. So, some of the hardest things are things like the pilus, right? The flagella and the fimbriae. Slime layer, you could also call that glycocalyx. That wouldn't be wrong. There are different names that you can use for certain things. Oh, okay, you're okay. It's okay. I might have to take a quick 5-minute break to get her happy again. So, know the different types of flagellar arrangements. Hey, it's okay. Let's stand up. Let's do that. Flagellar arrangements, right? That's going to be mono, lopho, amphi, and peritrichous. That was on the quiz. You might need to do something similar for the midterm, where you have to, there we go, standing up. I need to stand up anyway, right? Where you have to draw these out, or maybe you'll see images and you'll have to say, well, which one is that one? Where are the images, right? Maybe it looks like something like that. This first one is mono, right? This one is amphi to the right. This is lopho, a tuft at one end, and this is peri. No, compare and contrast gram-positive and gram-negative bacteria. There's not much about flagella, but they do move based on runs and tumbles. And runs are counterclockwise, and that goes straight. And then if they need to change their direction, they'll go from counterclockwise to clockwise, and that will cause a shift, a tumble. Okay? And this can be in response to certain chemicals and light. I'm going to bounce here, so hopefully that's not making you dizzy or nervous at home. Chemicals and light will either attract them or repel them, depending, right? Okay, periplasmic, that's the last flagella that's inside the cell, and it twists and helps it move, right? So flagella also helps with a number of different things like attachment and stuff, but really in this context, we're talking about them moving, like me, okay? Moving. Fimbriae, what do those do? Those are for attachment. Just know that. Know that they look like this, right? Fimbriae in the upper left-hand corner, and just know that they're for attachment. What else? Know the different bacterial shapes. Okay, so that's the one that really got a lot of you on the quiz. For gram-negative and gram-positive, you might have a question on the midterm that is like, draw a gram-negative cell wall and include the following things like porins and lipoproteins and phospholipids, right? It might be compare and contrast the cell wall of a gram-positive and a gram-negative, right? So when it says compare and contrast them, I'm asking you to compare and contrast them based on what makes up the cell wall, right? Gram-negative has an inner and an outer membrane that sandwiches that peptidoglycan in the middle, whereas gram-positive just have that really thick peptidoglycan on the outside. No outer membrane, no lipopolysaccharides, no porins, right? No lipoproteins, just lipoteichoic acid and teichoic acid. Right. Any questions on that? Any further explanation on what I'm looking for here? No? Okay. So then we'll get into shapes. Where are the shapes? Here are the shapes, right? Circular, coccus or cocci. Elongated, right? A rod or bacillus or bacilli. I don't really like you to use rod. I should just get rid of that. But cocci, coccus, bacillus, bacilli, if it's a rod shape. But I'd rather you not call it rod. And then if it has this curvature to it, right, the cheese puff kind of shape, then it's vibrio. Vibrio cholera is the example, right? It's not like a, I think of Good and Plenty's. I don't know if you guys have had those. My grandma used to always have them in black licorice, and they're covered with a pink or white candy shell. It's that shape, all right? It's a rod. It doesn't have curvature in it like a cheese puff. Why are you grunting? Spirillum, spirochete, and branching. Okay, for spirillum or spirochete, I will accept either answer. If you see this structure here in the middle and you call it spirillum, I won't dock you for that, right? But know that there's a difference between spirillum and spirochete and branching, right? Branching is not wiggly. Branching is, as it says, it has branches on it. Hey, why are you whining, okay? Pleomorphism, many shapes. The same bacterial species can have more than one shape, okay? And if those have arrangements that are not, we talk about strep as in a strip, streptococcus, staphylococcus as a clump or a cluster. Pleomorphic cells can have a palisade arrangement. So since they do have these different shapes, they can kind of, what you see here circled, they can line up and stack up like palisades, right, architecturally. So we don't have cocci palisades particularly, right? We have these pleomorphic shapes that are, again, going, where's pleomorphism? Right here, more than one shape. So it's not strictly coccus, it's not strictly bacillus. It can be those and anything in between. Be familiar with this kind of stuff, right? No coccus, bacillus, vibrio, spirillum, but then also know if there are two of them and they're cocci, it's diplo, not dicoccus, it's diplo. You need the diplo. Tetrad versus sarcina, right? A tetrad is just 4. Sarcina is 4, then continuing to divide into a cube. Diplobacillus or diplobacilli, again, 2 rod-shaped bacterial structures. Okay, so know these. This is diplo here. These were on the quiz. This one is strepto because it's a strip. Tetrad, 4. Sarcina, a bunch, right? Forming a cube. That's what I want you to know for chapter 3. Any questions? Shoot in the chat. I don't see any questions in the chat. Again, I'm looking for big picture concepts here. Okay, look back at those quizzes. Not because all the questions from the quizzes will be there, but it'll give you an idea of what I'm looking for with that material, and how I'm going to ask that from you. That's why we do these quizzes every week. The bane of your existence. All right. Let's push ahead. Let's do chapter 13, unless there are any questions. Okay. So for chapter 13, be familiar with antimicrobial agents' mode of action: cell wall, cell membrane, protein, synthesis, function, that kind of thing. So where is that? It's right here. Okay. And what do I mean by that? Okay, so the mode of action on a cell wall, right? You have things that break into that cell wall, and then it prevents it from getting things that it needs to maintain it, right? So we can prevent the cell wall from continuing to build on itself, which cell walls need to do. We need to add new lipids and new proteins all the time because they have half-lives, right? And they break down after a while. Another way to do that is to put in these surfactant molecules to actually dig into the cell and break it apart. And remember, that membrane is the protective semi-permeable barrier on the outside. If we break that apart, the cell dies. Okay, so that would be cell wall, cell membrane, protein synthesis, right, would be blocking the ribosomes, right? We had, sorry to jump around here, but there was, where was it? Did we get to that? Maybe that's on the, sorry, oh, that must be on the next, that's chapter 14. So protein synthesis, right, is going to be in chapter 14, blocking ribosomes from allowing the transfer RNAs to bring in the nucleotide, preventing the subunits from coming together, right? Preventing translocation of that RNA through that ribosome. You can prevent nucleic acid synthesis, and we talked about that more in chapter 14, specifically with the antivirals, right? We can have nucleotide analogs, right? They look like nucleotides and they can bind like nucleotides, but then it prevents the DNA from replicating. Other ways is we can use chemicals to mess up the shape, because shape is function, right? So we can completely denature a protein, take away its shape entirely. It's not going to do its thing anymore, right? We can change it to a different shape. So we don't denature it, but we change the shape sufficiently enough to where now it can't bind and do its activity, right? Or finally, we can block this active sites on the protein, right? So we don't change the shape of the protein, but we use competitive inhibition to block the sites that it uses to take care of its business, right? And now it can't do its business, and so the protein is functionless. So there's more than one way to skin a cat. All right. And if you like cats, sorry for that expression. I don't have anything. I've never skinned a cat. I don't have anything. I have dissected a cat in college, but I don't have anything against cats. What else? Be familiar with the different methods of control. All right, so let's go back. Sufficiently hydrated versus dry, right? Autoclave versus incineration, right? Cold temperatures, desiccation, those things are going to be microbiostatic, right? Not microbicidal. Cold temperature and drying out doesn't kill the bacteria per se. It prevents them from replicating, makes them unhappy. Be familiar with these terms. What's the difference between sterilization and disinfection? Well, they're both used on inanimate objects for the most part. Sterilization kills everything, spores, endospores included, whereas disinfection is only good on the vegetative pathogens, not endospores. Antisepsis, that's applied to body surfaces. So that's how that is the disinfection that, equivalent. But if we're decontaminating the surface of our bench, that's disinfection. If we are decontaminating the surface of our skin, that is antisepsis. And all three of these things are microbicidal, whereas cold and desiccation or drying out are microbiostatic. All right. Some more things, sanitation, right? Inanimate surfaces. Degermation, living surfaces. High, moderate, and least resistant. You don't really need to know those as much as you need to know high, intermediate, and low level germicides. And there is a lot there. So what I want you to know, and that's getting into toward the end, just give an example of high, and just know, for these different germicidal categories, right? No, alcohol, low or intermediate or high, right? Heavy metals, is it low, intermediate, or high? Okay, that level of resolution. Ionizing versus non-ionizing radiation. All you need to know is that ionizing radiation breaks DNA. Non-ionizing radiation causes pyrimidine dimers, right? It causes those nucleotides, those thymines, to form a bond together. They dimerize. And so now you can't translate that, or you can't transcribe it. And so it prevents RNA synthesis and downstream protein synthesis. Liquids. If you're using it on, again, living things, it's antisepsis. If you're using a liquid on an inanimate thing, it's either disinfection or sterilization, depending on the properties of that liquid. Yeah, well, you want some coffee? No, not going to get coffee. Thermal death time versus thermal death point, right? The amount of time for TDT is what that definition is. And then TDP is the lowest temperature required. Okay. Those are easy to mix up. So for this stuff, right? Again, on the quiz, I wasn't looking for you to give me all the details, 15 PSI, 121 degrees Celsius, the time, right? Know that autoclave sterilizes. Right? Know that tyndallization also sterilizes, but it's an intermediate pulse of steam, of heat, right? It's non-pressurized. So it is still a sufficiently hydrated heat control. Boiling water, that is a heat control with moist heat, but it doesn't sterilize. Pasteurization, right? Just the basic details. What's the matter? Yeah, okay. Incineration, that sterilizes, right? That's why we flame from loop to stem, right? Because we are incinerating that stuff. What are you at the light? With this light? Here. There you go. Play with that. Play with the light. All right. Again, terms you'll want to be familiar with: microbiostatic versus microbiocidal. Non-ionizing versus ionizing radiation. And then high, intermediate, and low. And that's what I say on that last bullet point for chapter 13. Be familiar with the different germicides and whether they're high, intermediate, or low. So with these, it would be halogen, and you would say that's intermediate. Okay. You could say chlorine and iodine. You could give more examples, but really being able to just recognize these different categories and the efficacy, right? Yes, it will be important to know, do I use chlorine on a patient? Well, no, of course not. I can use iodine if they don't have allergies, right? Phenol, low to intermediate. Yes, it is used on people, but it's questionable nowadays. Really what you need to just be able to recognize is it's phenol, low to intermediate. Okay. Alcohols, right? Those are going to be intermediate. Hydrogen peroxide, that is going to be, oops, don't have it on here, I guess, but that is going to be intermediate. Aldehydes, high level or intermediate, depending on which aldehyde you're using. So you could say intermediate to high. What? Yes, you got that. Boy. Yeah. Gases, high level, right? That's what we use to sterilize things that won't survive in an autoclave. Right? And survive, I mean, they won't withstand the temperature and pressure and maintain their consistency, right? So that is a way to sterilize when you can't autoclave something. All right, detergents and soap, low level. You don't need to know structures or anything, but that heavy metals, low level. Dyes, low level. Okay. So it might be, some question like, name as many germicidal categories that you can, and let me know if they're high, intermediate, or low, or a combination thereof. That's all I'm looking for. Okay, you don't have to talk about acids and alkalis being low and talk about propionic acid and benzoic acid, okay? Just know that those are low level. Any questions on this? No questions. Okay. I'm just going to keep moving. Hopefully this is beneficial to you guys. What do you think? You think this is helpful, or are you just being a distraction the whole time? Oh, no? Okay. All right. Chapter 14 then. Here we go. Moving forward. What time is it? I'm going to keep going since we go into lab and stuff. Like I said, I am recording this, so if you need to take a break or something, that's fine. You can pick it back up when I post this to Canvas. Okay, so chapter 14, be familiar with the 5 major components that are useful drug targets and provide examples of respective drugs and whether they are broad, narrow, or broad or narrow. That seems like a lot, and it is kind of a lot, but it's not nearly as much as if I had you memorize all of these things. So the major targets are going to be cell wall, nucleic synthesis, DNA or RNA, right? Plasma membrane, ribosomes, and metabolism. Those are the 5 major areas that are targets. Give example, right? So for cell wall, you could say beta, you could say beta lactam, right? Is beta lactams like, or you could say penicillin, right? Penicillin. And you could say penicillin's actually broad and narrow, depending on which one you're using, right? So be familiar with these 5 categories and be familiar with an example of a broad and an example of a narrow. And if they don't have one of those, just be familiar with one that they do. Okay, you don't need to memorize all these drugs. You're welcome. So again, review these. This is one. This target is on the bacterial cell and penicillins, right? Broad spectrum. Could also be narrow spectrum. Right? If you want to use a non-beta lactam, you could say vancomycin, narrow spectrum, right? Or bacitracin, narrow spectrum. For disrupting the cell membrane, you could say polymyxins, right? And you would say whether or not that's narrow or broad. In this case, polymyxins are narrow. So maybe you make a flashcard and you have the 5 different categories on there. And then on the back, you have an example and whether it's broad or narrow. Oops. Okay. Nucleic acid synthesis, right? Again, antiviral drugs are analogs, are examples of that. And those, those are going to be, depending on what antiviral you're using, it could be a pretty narrow spectrum. Here's a fluoroquinolone that is a broad spectrum, right? So that would be an example you could use for a broad spectrum that works on DNA synthesis. Protein synthesis. You can use streptomycin, gentamicin, tetracyclines as examples. You don't need to know all of these though. Okay. Hey. Likewise, I'm not going to have diagrams here and say, well, what drug is this and how is it acting on this ribosome? But be familiar broadly some of the different ways in which we interrogate ribosomes. And remember, their ribosomes are different size than ours. And that allows us to be specific to bacteria and not hurt our own ribosomes. Right? And we can have the mRNA misread. We can have the peptide being blocked because we aren't allowing the amino acids to get in there properly. We can prevent the 2 subunits coming together. What's the matter? What's wrong? What's wrong? Right? So there's these different strategies. And again, you don't need to memorize the drug that's corresponding with each of them. Maybe be familiar with just loosely how we do that though, in terms of what are some different ways that the ribosome can be essentially prevented from doing its job. Okay. And then 5 mechanisms of acquired drug resistance. So again, this is information that will be helpful, but not super essential, right? If it's not on that outline here, it shouldn't be on the test. So here's drug resistance. drug inactivation. That's one mechanism, right? So we change the structure. We have an enzyme that we produce, we being the bacteria, that inactivates that drug because it changes its structure. You don't need to know the structure of penicillin. You don't need to know the bond that it disrupts, but just know that the bacteria changes that drug. Another one is decreased permeability, right? So we change the receptor shape that is normally used to internalize that drug. So now the drug can't get in there anymore. Drug pumps, right? That's feeding the baby. Food goes in, food gets spit right back out. Okay, so they activate these drug pumps. Any drug comes in, it's recognized as being harmful, it just spits it back out. Change in the drug binding site, right? So in this case, particularly, it's a ribosome and it has a triangle shape. Well, we change that shape to be circular. And so now the triangle drug can't bind to it, can't fit in there like a lock and key. And then finally, alternate metabolic pathways. So if we are doing something to disrupt the production of folic acid, for instance, by perturbing a step in that pathway, it can evolve alternative pathways. Mama. Okay. All right. That's it for 14. Okay. Any questions on 14? No? Okay. I realize this is less than ideal. We'd rather be doing this in person, right? But it is what it is. So we're going to move forward. And again, please feel free to ask questions now or later if you can't think of any right now. But, okay. I'll put you down. Let's do this. Let's take a 5-minute break. Okay, so and then at 10:10, I'm going to come back and she's not going to be crying anymore. I promise. Kind of. No promise. So I'm going to be back soon. All right, I'm back. I'm giving Nora some Nutella, and I've got it kind of in a cup. I'm hoping it'll work as like a Kong. I don't know if you know what those are, but it's like a toy for a dog. You put peanut butter inside and it occupies them. So don't judge. Nutella for breakfast isn't the worst thing, right? All right, so chapter 15 and 16, the last bit. This is the long list. It takes up an entire page, right? That said, it is 2 chapters. Yeah, you've got some in here. Look, that's what this is for. You get it out. See? So major factors in development of infection, right? That's going to be here. Okay. Know those. Now we go into a lot of detail with that, right? But I really want you to know those 5 more so than the detail, right? Because if you know finding a portal of entry, that's pretty self-explanatory, right? Attaching firmly, yes. You could tell me what it uses, the different structures, and there's a whole table with different microbes that use different things to attach themselves, right? But for the midterm, I really want you to know these 5 and the sequence that they're in, right? Don't say attach firmly and then find a portal of entry, right? It's portal of entry, then attachment, then survival, damage, exit. Be familiar with some factors that weaken host defense and increase susceptibility to infection. That's going to be things like age, right? Elderly, young. She's like been sick, I don't know, continuously for a couple of weeks now. It just goes from one thing to the next, right? Her immune system isn't as mature as ours. Being immunocompromised, right? Whether it's from cancer and having radiation and chemotherapy, or if it's because you have an autoimmune disorder and you are on immunosuppressants, right? Or if you're just infected with something else, it makes other bugs more likely to gain entry and take up residency as well. Okay. Compare and contrast true versus opportunistic pathogens. Where was that? I think that's further back. It's in the slide deck, but true pathogens will infect anything regardless of their immune system. They're in the business to make you sick. They're in the business to cause damage. And so a true pathogen indiscriminately infects hosts. An opportunistic pathogen takes advantage of a situation to become pathogenic, but it is not, doesn't have to be pathogenic. Okay. It will only be pathogenic if the host is immunocompromised. Okay. And that could be immunocompromised because of cancer or because of an autoimmune disorder or because it's being infected with something else, right? AIDS patients, they're immunocompromised because they're infected with something else that also happens to attack their immune cells. But Nora here, she's perfectly content eating her Nutella breakfast, and she's sick with something, and her immune system's on alert and fighting that off. But in the meantime, she could get sick with something else. Opportunistically, that's not a pathogen otherwise, okay? So that's the main difference between true and opportunistic pathogens. Compare, well, they're both potentially pathogenic. They can both kill or make someone sick. Next, know the difference between these things: signs and symptoms, incidence, prevalence, mortality, morbidity, pandemic, epidemic, right? That's kind of what we wrapped up with at the end of the lecture. Right, so a sign is going to be something that you can empirically assess as the outside observer, right? Fever, cough. You can take draw the blood and look for signs of lowered white blood cell count, right? Or sepsis. Yeah, symptoms, right? General malaise, right? Feeling bad. Well, if you go to the doctor and you say that you talk to the medical assistant or the nurse or the physician and you tell them I feel bad, that is a symptom. Okay, and that's not to say that symptoms aren't helpful, right, in diagnosing, but just in terms of vernacular, they are different. So there was that table. Where was that table? The table right here. That's the one table that will you'd benefit from being familiar with. Okay, because I might say leukopenia is a sign or a symptom, right? Well, you kind of got to know what leukopenia is to know that, right? But generally, someone doesn't say, "Oh yeah, I feel terrible. I have leukopenia," right? It's a sign. Incidence versus prevalence, right? That's coming here. Incidence is the number of new cases. Prevalence is the number of existing cases. So how many people to date? Don't, what, do you want more? You're just like hitting me with your spoon. How many people to date have this particular disease? How many new cases per week or per month or per year? It depends on the metric they're using. Mortality, death, morbidity, just having that disease. Mortality, the number of deaths, right? So COVID, we break it down to mortality. How many people have died from COVID versus how many people have had COVID? And depending on the pathogen, those numbers will be similar or dissimilar. We hope they're dissimilar. Pandemic is a pathogen that has spread around the globe, whereas an epidemic is still contained within a geographical region. So that's like the meat of 15 and 16. And then there's like this long list of definitions and terms, okay? And so, we can go through those if you want. I think they're pretty self-explanatory. Oh, you're all done? You went out? Look at you, you're a mess. What am I going to do with you now? I don't think I'll go through all of these because I feel like I said, exogenous agent versus endogenous. Exo is outside, endo is inside. Okay, you went out? Yuck. Okay. So I think from there, I'm going to just open it up to questions. If you have any, right now is the time to ask. If not, like I said, throughout the day, feel free to shoot me some questions. Okay. I know that you have a midterm coming up, some of you before this, and you probably are anxious to start getting prepared for that. But I'm happy to stick around and address any more questions as they come. Look at this. Yeah, that Nutella all over you. You can feel free to ask, shoot out loud. Or send me a chat. And if you don't have any questions now, feel free to hop off and, pick up with your day. No lab today, so you have a couple extra hours to prepare for whatever, whether that is for your A&P or for your micro, or if you just need to go for a walk or a bike ride to clear your mind. You have that carved out. Okay, great. All right, well, I guess I will end it here. Once this gets done processing, I'm going to post it to Canvas we're in week 6, right? So it'll be in this week's Canvas. Again, if I didn't cover something that you had a question on, feel free to shoot me a message. And if there's any way that I can hop on Zoom and talk it out, I'm happy to try and do that as well. Okay. I'm just going to hang out today with Nutella Girl. And so I hope that this was, again, helpful and that it wasn't too much of a distraction. And I look forward to seeing you on Wednesday, and, hang in there until then. Okay. See you later.